And welcome, ma'am. Kindly deliver your talk.
Okay, thank you very much. And so, as we said yesterday, today I just complete, let's say, the section of about the basics of fluorescence spectroscopy, and the topic is FRET, and that's very important methods with several type of application, and so then it's important to say some words around. So, what is FRET? FRET, so then is an acronym, and if we limit now on the last three letters, so RET, and so it stands for resonance energy transfer. So it's a mechanism describing the energy transfer between two light-sensitive molecules, so two chromophores. That is a very general. And so there's also as historical, so there are of course, some historical news. So then was first observed in the beginning of last century by Cairo and Frank, who observed that the excitation of a mixture of mercury and thallium atomic vapors with the 254 nanometer, the mercury resonance line, display thallium emission at 535. So you see, that is the importance element that we will see. It's recurring in this kind of method. So was excited one chromophore, and the emission was from the other one. Then several other scientists contributed to better understand this phenomena, till arriving to the work of Förster, who was in the middle of the last century the developer of the first quantitative theory of molecular resonance energy transfer, which we still use today. And so then we arrived to define the F in this acronym, which initially was considered to be stay for fluorescence. But in reality, fluorescence is not really involved. Also, it's not the central phenomena in this in FRET. And so then, more recently, it's even more and more common to use F for Förster, since is really indicating actually the type of quantitative theory that need to be used to analyze FRET. And so let's see why I say that fluorescence is not really involved. So, okay, that says the observed phenomenon. So we have a donor, a molecule that we call donor, and a molecule that we call acceptor. So what happens? We excite the donor, then we observe the emission of the acceptor. And what happens in between? Is this energy transfer. And that can be radiative. So that means that we can imagine that there is the donor is excited, then it emits, and the wavelength emitted by the donor is absorbed by the acceptor, which is excited, and so it can emit. So that's one would consider. So that's, but can also be non-radiative. So that means that the donor is excited, and the acceptor emit, and the transfer is made by resonance. So in this case is non-radiative. So that's why, and the resonance energy transfer is this second phenomenon. That's why fluorescence in principle is, that's why fluorescence is not involved, because the donor It's not emitting, so it's not the fluorescence of the donor that is reabsorbed, but the transfer of energy is done by resonance. Okay, so what are the characteristics? So then, so in radiative energy transfer, is in, if you want to, in reaction scheme, what happens is the following. So then we have a donor that is excited, which go back to the ground state and it emit a photon. And this photon is reabsorbed by, is absorbed by an acceptor, which get excited itself, or in case, sometimes if there is an overlapping of the spectra, is by the donor. And in this case, will be homotransfer, because it's transferred to the same molecule. So yes, I put already also the characteristic. So in the spectral characteristic. So in non-radiative resonance, instead, as you see, so there is a transfer, so not the photon here, it's not appearing. So there is the donor that is excited and together with a ground state acceptor, and then we have the donor in the ground state and the acceptor in the excited state. In case of heterotransfer, and among two donor molecule in case of homotransfer. What are the spectral characteristics that can tell us if we are in one or in the other situation? So in case of radiative, the emission spectra of the donor is modified. So the intensity practically decrease in the region of spectral overlap, because actually the photon emitted in this region by the donor, they don't reach the detector because they are absorbed again. So to produce then the emission of the acceptor. So that's is the major difference. The fluorescence decay instead is unchanged in case of heterotransfer, because as we said yesterday, lifetime is an intrinsic it's an intensive parameter. So in case simply we practically reduce the concentration of the donor, but the donor that emits anyway, and so since the lifetime does not depend on the concentration, so the lifetime practically stay the same. The donor. In case of non-radiative resonance, instead, this spectra modification is not observed. But what is observed is a decrease of the donor emission, because the photon, so it's not the photon, so I mean due to the resonance, the concentration of the emitting donor is decreased, because it goes back to the ground state, and so it's no more excited, but it's goes back to the ground state without emitting a photon. So there are less photon coming from the donor. But the donor that is able to emit, because it's not transferring its energy to the acceptor, emit in the normal way. So then it's just, we see a decrease of fluorescence, but not a modification. And in this case, the fluorescence decay instead is shortened because the donor stay less. So the energy transfer is practically, in this case, is a non-radiative pathway that is added to all the other non-radiative pathway that reduce the time the probe stay in the excited state. Okay, so exactly. So that's what I was saying. So coming back to the diagram of Jablonski that we have already seen. So then here, for example, just I show you yes. So the orange is our fluorescence signal, as we see. So the only one with the emission of a photon. Then we have all other different pathway, different type of quenching, internal conversion, and so on. And the energy transfer is part of this non-radiative pathway. So it goes to make the rate constant K and R more important. So this is what happens then. So in this case, I reduce the diagram, the Jablonski diagram of the donor. I make compact. So the blue is the fluorescence, and with the gray arrows, let's say the non-radiative pathway altogether. So that is the two element of the emission. That is the donor alone. When I add the acceptor, so they can be part of the excitation is transferred to the acceptor, which it found in itself in the excited state. That is with resonance. And so you see there is another pathway of this excitation, and I took out some arrow from here to put this one. So and then there will be the emission of the acceptor, and this transfer. So here is the KT, the rate constant that goes to increase the non-radiative rate constant. And this transfer is done by resonance, like in acoustic. That is we have to see this idea. So there are the dipole that are getting in resonance. Okay, so that's the effect. Reduce, since KNR is larger, the quantum yield is reduced and the lifetime is reduced. Okay, so these are all the parameters that is that enter, let's say, that play a role in the transfer rate constant. So let's see the physical parameters that are involved. And so we have so that we will analyze and then one by one. So this part here is the contribution of the overlapping between the emission of the donor and the absorption of the acceptor, because that is something that has to happen. This R instead is the distance between the donor and the acceptor, which is also play a role, because closer they are, easier is, of course, to enter in resonance. Then we have the others parameter that were the physical parameter more yeah, let's say. Okay, so other parameter that play a role is the quantum yield of the donor without acceptor. Then there is the refractive index of the solution. Okay, this is the lifetime of the donor without acceptor. And okay, this is the number of Avogadro, and all the other are numbers that are due to the mathematical model to solve this kind of equation. Okay, so once we have the KT, if we can rewrite this complex expression in a more compact form, where you recognize some element, in particular tau d and r, and all the rest is included in the R0, as I've indicated here, which is called the Förster radius or critical distance. It has the dimension of a distance, and is called the Förster radius. So that is practically the result of the calculation of Förster. That's why it's called like this. Good. So few physical element to see to explain the interaction energy between donor and acceptor. So some little quantum mechanics view. So when we have a situation like this, so the initial, that we can call it initial state and final state, and in waveform, that is the initial state waveform. So then you see here we consider a 2-electron system, and that's so because the donor is excited and the acceptor is in the ground state. And that is the final state waveform, where the donor is in the ground state and the acceptor is in the excited state. So what happens? You see, there are two ways to transfer the excitation between the two molecules. So one is called the Coulombic mechanism, and the electrons move between orbitals of the same molecule. So that means that you see, so you have the electrons that is in the excited state here, that goes down, and due to this resonance, the electron that is in ground state of the acceptor goes to the excited state. You see, follow the 1 and the 2, which is expressed here. The exchange mechanism, instead, electrons migrate from one molecule to the other. And so then it will be practically the electron of the donor that is in the excited state goes directly to the excited state of the acceptor, and the electron of the acceptor, which is in the ground state, it goes directly to the excited ground state of the donor. So back and forth. So that's are the two effect. Okay, so yes, in the calculation, if we want to calculate the interaction energy, then we have the total Hamiltonian, where we have of course the Hamiltonian of the donor, the Hamiltonian of the acceptor, and the interaction energy is introduced as a perturbation. So the coupling between initial and the final state is represented by this matrix element. So let's not enter into the details of quantum mechanics, but it's interesting to see from where actually common equation that we are using every day, from where they come from. Okay, so this matrix element, has this two contribution. The two contribution of the Coulombic part, which and of the exchange part. So then, if we use this part of this other part of the wave function. Okay. So yes. So the Coulombic part can be expanded into a multiple series, because and the dipole approximation donor acceptors, and practically what happens, we have to consider that we have always said that the molecule are represented in spectroscopy, are represented by their corresponding dipole. So we have the dipole moment. In this case, what are the dipole involved is the dipole moment of the transition d star in the donor, and the dipole moment a star in their acceptor. And so what happens is practically this, that you see you have a dipole field, and the dipole field around the donor is decreasing to go to enhance the one in the acceptor field. And so in the dipole approximation, donor acceptors, when the donor acceptor separation is much larger than the molecular dimension, the dipole potential, let's say, we can simplify to this cubic term. And voila. Okay, so in the exchange term, instead, represent the electrostatic interaction between the charge clouds, and so is actually short range. So you see here we are in something that, as in the Coulombic term, can occur at a larger, and actually when the donor acceptor separation, so that means the separation of the molecule is larger enough and is larger than the molecular dimension. So we can have this approximation. Instead, here to have the exchange term, we need really the overlap between the charge cloud, and so then has to be short range. And the profile is more exponential. So that means that, so you have these two terms, the Coulombic interaction and the exchange interaction, and we see that actually in the FRET, as we normally consider, we are practically consider this dipolar approximation, and that is what FRET is. So the forced resonance energy transfer, where instead the excitation exchange term is playing a larger role. Instead, there are some short range effect, and in this case, the model of Foster is not really working. And so that's actually, so it's not all the time that we see the emission of the acceptor when we excite the donor. Is not, we are not necessarily always in the Foster condition. So we have to be careful and not simply use equation just because it seems that they are okay, but sometimes we have to first be sure that we are in the right condition. And we focus on the long-range effect, which is the threat properly said. So yes, okay, exactly. That's what I want to say. So that is in the yeah, maybe the animation was too slow in this case. So this is the what happens in for the Coulombic term. So then we have interaction. That's why Coulombic, because it's electrostatic. So we consider just two dipole. And instead, in the exchange term, we need really the overlap of the electronic clouds. So then, of course, the molecules have to be closer together. And that's our known threat phenomenon. Good. So then, as a yes, just to see then from where the equation come from. So to introduce practically the interaction dipole that we have just underlined in this in the rate is so using this. So I just make everything. So is let's say, so this cubic term that is part of this dipole-dipole interaction in the square mode due to the Fermi's rule, which is giving us the transition rate between the two states, the initial and the final state. It's provide this R power 6 that we see in the rate. Okay, so then let's say that from all what I said, and now we keep just with this equation, which is not that this expression is not that is simpler, is simply I just show you from where it comes from, this power 6, which is which is typical of threat. So now for all what was said, you can, let's say, practically almost forget everything, but just remember that the fundamental aspect of this type of transfer is due to the to a dipole interaction, and we are is a long range effect, and that's his threat. Okay, so the orientation factor, which is this factor that is indicated here, is also come from the dipole-dipole interaction potential, and because here, if you solve all these scalar product, so then you have all these cosines that will appear, and that is the expression of the orientation factor, which of course is related to the orientation between the dipole, and we will see it. Okay, yeah, where every time I move this part, it's okay, why? Yes, so the Foster radius, as I was said, yeah. And now let's come to something more practical. So, okay, so more visual. So what is the overlapping integral? The overlapping integral, as we said, so in order that the transfer occur, so now more practically, we need an overlap between the emission of the donor, which is here in green, and the absorbance of the acceptor. These spectra that are collected here are taken in absence. So when that is the donor alone, that is the acceptor alone. That's their characteristic. And one see that there is actually an overlap. and so on. So the overlap is this one. And and we can calculate, let's see if, yes, and we can calculate the area of this of the overlapping region between the fluorescence spectrum of the donor normalized on the wavelength scale and the extinction coefficient. So that is a parameter that informs us on the emission, and that's, of course, is a parameter that informs on the absorbance. Okay, so this area need to be calculated, and these are the unit involved, and we will get this overlapping integral here. So in one experiment, so this is what we practically obtain. So yes, that is. I have something that is some technical that I have to move because it covers my.
You can hide. There is a stop sharing. There is one small button. Yeah, you click it.
Okay, yes, okay. So that's is. Yeah, okay, good. Now I see. Boom. Okay, thank you. Yeah, okay, we are in the situation. Green is the donor, red is the acceptor. That is the interaction between the two, and we can calculate the FRET efficiency. So as every efficiency is calculated as a ratio of the process of interest, KT in this case, on all the others, and so if so since we know now that the KT is expressed in function of the distance between the chromophore, and that is the definition of the fluorescence lifetime of the donor. So, and if we introduce this expression we combine them, we can pass from the efficiency expressed in function of the rate constant to the efficiency expressed in function of the distance, which has more practical, let's say, utility. And okay, so that is a FRET experiment. So sorry, I was making. So that's a FRET experiment. So that's let's say are the characteristic of the donor and the acceptor. And when they are close enough, transfer occur. And you see the fluorescence of the donor decrease and the fluorescence of the acceptor increase. So, okay. And we can actually characterize the efficiency of the transfer also in this way. Because if we practically here, in this way, we quantify the fluorescence that is how efficient actually is the transfer between the two. I compare the fluorescence of the donor in presence of the acceptor to the fluorescence of the donor alone. And so what is lacking is the fluorescence, let's say, the fluorescence that cannot be seen because there was energy transfer. So that is the practical way of measuring the efficiency. And with the lifetime, what happens is the following: as we were seeing, in presence of the acceptor, the lifetime of the donor decrease, is getting shorter, because we have this additional decay pathway, and again we can estimate the efficiency, and then evidently, so this is the way in which we measure the efficiency of transfer, and then we can then translate this efficiency in an information on distance because of this relationship. Okay, just what I was saying, just to say how is the orientation factor calculated. So it play a role in the critical radius, the Forster radius here. And we saw that was this combination of cosinus of the angle that are described here. So in blue, you have the dipole of the donor, in red, the dipole of the acceptor, and that all the different angles are defined. So the theta D and theta A are the angle defined between this dipole and the distance vector between the two chromophore. And instead, the theta AD is the angle between the plane containing the dipole. And we can see that considering all the configuration possible, the orientation factor is between 0 and 4. And in particular, 4 is when both transition moments are in line with the vector. And 0 can occur actually in several ways when, for example, transition moments are perpendicular to one another. Okay, good. So, in case, so it's of course it's a little bit difficult to estimate the orientation factor, because we cannot go to measure all the angle between molecules, especially in solution that they have isotropic orientation, let's say. So in general, in our experiment, we consider the isotropic motion in solution, and so we get a dynamic average, and the value of the practical value of the orientation factor that is often used is 2/3, the dynamic average. But just to say that this parameter anyway has a meaning, we can see this in some more rigid system. So then let's consider a molecule of DNA. So so there is the double helix and the base pair, the pair of bases are oriented just a little bit. The orientation is changing in this double helix. Okay, so then this was an interesting experiment that was done. So that is DNA labeled with the two chromophore, a donor, two cyanine dye, cyanine 3 as a donor, cyanine 5 as an acceptor. Okay, FRET can be measured, and FRET was actually measured in function of the length of the DNA. And of course, if you change the length, so the number of practically of base pair, and so the different number of base pair means different orientation, because you practically you go up, and every time a step it means another orientation. And so this orientation was, of course, reflected in the orientation between the two dies. And very interesting, it was observed that the efficiency, okay, so let's say that the efficiency of transfer of between the two dies was modulated according to the in function of the number of duplexes. So practically was modulated by the orientation between the dies. Considering that theoretically we know how the orientation factor is defined, that was the simulation, that was what was expected, considering that we know also how the orientation are in a DNA molecule. So that was and the simulation, what was expected, and that is the experiment. As you see, the modulation is quite respected. So the position of the maxima and minima of the experimental profile are consistent with the modulation due to the orientation factor. And so that means that was a way to see that is experimental evidence of the meaning of the orientation factor. The isotropic average, as you see, would have not really well explain the effect. So it's a little bit, so of course the way is more or less, so it's, the tendency is there, but it's much more flat. So here to use an isotropic to use the dynamic average of the experimental of the orientation factor would have not be really correct. Yes, okay, good. Let's come to some some important consequence of measuring fret. So actually one practical reason why we make fret experiment, but the practical reason is exactly this distance dependent. And so, as we said, as soon as we can measure spectroscopically the efficiency of transfer, as we show, we have an information about the distance. And in particular, normally what we are interested on is this small r, lowercase r, that is the distance between the chromophore. The R0 instead is something that depends of the characteristic of the two chromophore and can be calculated in advance. So it's a characteristic of the pair. So it's something that is in principle is known. Okay, so then I show everything. So good. So that is practically as every efficiency. So it's something that is a parameter that is between 0 and 1, and of course larger is the distance between the chromophore, so the R, this R here, this R, sorry, R. Lower is the efficiency at certain point is 0. So, and of course, if so, as this is, and in this graph here, I try to highlight the confidence range. So practically where this equation, let's say, work at best, because you can imagine that it's difficult to distinguish value in this region and also in this region. So here, because it's actually quite flat, the change are minimal, and then here also. So that's you. So that means that a fret pair has to be chosen in order to have a R0 that is in the range of the distance that we want to measure. So that is the confident range, practically. So here an example of some fret pair. And so the donor here would be the tryptophan, and the acceptor is called uncial. And considering their characteristic, the quantum yield, the overlapping integral of their spectra. So we have a R0 of 21. So that means that if we use this donor pair, the donor acceptor pair, we can measure distance between 10 and 14 angstrom, practically. If in case we need to measure distance that are larger, we know this, that we want to, so then we have to use another fret pair, for example, for essays and tetramethyldrhodamine. Okay, so fret can be used to provide us structural information, as we see, since we can measure distances, but we can also study molecular interaction. In this, for example, so that's more schematically, so let's imagine that the donor is on a receptor. When we let a ligand, which is a fluorescence, which is fluorescent too, and it contains the acceptor. So you see, then the interaction is monitored because the emission of the donor is decreased and the fluorescence of the acceptor appear. The ligand could also be an acceptor that is non-fluorescent, so is a quencher. So in this case, the signal that we will follow in this kind of experiment is simply the decrease of the fluorescence of the donor. Yes. We can also study dynamics of biomolecule. So let's imagine to have the donor acceptor in two regions, each one in one domain, a different domain of a macromolecule. And that's what would happen as a consequence of the movement. So you see, as soon as the acceptor gets closer to the donor, we have more and more this, we can monitor the fret, monitoring the decrease of the fluorescence of the donor and the increase of the fluorescence of the acceptor. Another application of fret are in fluorogenic probes. So that is something that is also linked to the next topic, that is talking about probes in general. So yes, so you see that that is a probe that was designed to monitor cleavage reaction. So that was the first one that was the first time this method was used on a substrate of on a substrate for a HIV protease. And so how it's working practically is depicted here. So you have your substrate enzyme that will be here, and that is in the case of this substrate is the peptide that containing the cleavage site of this protease. And you have a donor and a quencher, also dark quencher, actually. So that means that when you have in the closed form and the substrate is not cleaved, you don't detect any fluorescence because the fluorescence of the donor is fully quenched by the presence of the acceptor. Then the enzyme come, cleave the bond, the peptide bond, and the two moiety get separated, and you can see the fluorescence of the donor. And so you monitor the cleavage reaction. Can be used also for nucleic acid recognition. That is the scheme of one application of the molecular beacon that are such a type of probe where you have also a fluorophore donor close to a quencher. So at the extremity of this stem loop structure, and when it interacts with the complementary sequence, so the stem is opening and hybridized to the complementary sequence, and as a consequence, the donor get farther away from the acceptor, and so it's not quenched anymore, and we can monitor the restore is fluorescence, and we can monitor this reaction, and so detect this particular sequence. That is actually, I said, a different issue. So I just say that maybe someone of you could also say doubt that since here the molecules, see, they are even presented in contact to each other, and they say, okay, are we sure that we have a really threat? In fact, that is also a point. Sometimes there are we see something that looks like threat, but actually is not really threat. And when the molecules are very too close, there are some phenomena of static quenching that are involved. Threat, in fact, the real threat is a dynamic quenching mechanism because it involves the excited state of the donor and then acceptor, and they don't imply any modification of the absorption properties. Instead, static quenching, as we talked yesterday, involves the formation of a ground state complex between the donor and the acceptor quencher. And that's as a modification of the absorption properties. And this was also actually what was observed in some structure like this, so it was observed that when the spectra of this molecule with the fluorophore and the quencher on it, has a different absorption spectra in comparison to an equimolar solution of the single labeled structure. So meaning that when they are too close, the absorbance is changed. So something happens. So there is not fret, or at least there is not only fret, there is also something else. And so that was also a work that was done some time ago in the lab where I am now, supervised by Professor Meli. And so taking care about this phenomenon was allowing to better quantify the dynamic of opening of this probe, also in yes. So then maybe yes, I show it here. So that you see when you want to follow. So that was the real system on which it was used. So if you want to solve the opening, the annealing process, so you start from the closed form, and then step by step you open the stem, and to take care about also static quenching phenomena, introducing this in the analysis, it was possible to inspect also the early step of the annealing, because it's exactly in the early step where the quenching, the static quenching phenomena, and so the exciton coupling that is occurring, so is more important. And then as soon as they get far apart, so we pass from short range to long range effect, and then we can continue using the fret model. So that is an example of how that equation has to be not just used blindly, but has to be sure to be in the good condition. Okay, so I guess that, yes, that ends an overview on fret. And in case if there are urgent questions, I'll be happy. If not, I will move to the next session that will be on fluorescence probe in general. So, is okay? Are you all there?
Yes.
Yeah, okay.
Yeah.
So, but then, but let's move on. Okay. So, okay, following the plan of the schedule, then we go to give some general concept about fluorescent probe. We talk already about some of them. Now let's make a sort of X-ray or on the important fundamental characteristic of fluorescence probe. Well, we know now fluorophore, that is a molecule or a portion of a molecule that has the ability to emit light upon excitation by a external energy source. And so a molecule that is fluorescent practically. And about fluorophore, we can distinguish two major categories. We have intrinsic fluorophore, which are a naturally occurring probe, and extrinsic fluorophore, which can be synthetic dyes or modified biochemicals, so the analog, that are added to a specimen to produce fluorescence with specific spectral properties. So here are some examples of intrinsic biochemical fluorophore. So for example, in protein, there are the fluorescent amino acid. Tryptophan, we have already talked about it. Also tyrosine, phenylalanine, a bit less. It's, as you see, is more spectrum, more in the even more in the UV range and very low signal. Okay, so then there are also some natural cofactors that are fluorescence, like FAD or NADH. And then there are a lot of, so of course there are, and then there are all the extrinsic fluorophore. I mean, here we have a lot of type of dye, so that is just some example of dye that maybe you heard already about. Then just yeah, here also more recently, even if now is less and less recent, was a development of these Alexa dyes that you see the interest that is really a work to go to develop a dye, and this was really done systematically to try to hit all the wavelength. The numbers that are written here are the maximum of absorbance and the maximum of emission. That gives us an idea where these dyes actually may work. And so you see, it's just modify some group, you and in particular, as we were saying the very beginning, increasing the degree of conjugation, you move to larger wavelength. And there is actually really a need of long wavelength probes, and in particular with the smaller size, because in fact to reach long wavelength, you are obliged to, you can do this actually by increasing the conjugation, the degree of conjugation, and so then you have a tendency to increase the dimension of the system, as you see here. And that's may have troubles because you get the probe that are larger and larger, and sometimes you don't want. So then it will be nice. I don't know if you also you want to work in this domain. So yes, that's is always. The community is always interested in long wavelength probes, but with small dimension. So, okay, then beyond organic dyes, there are also other extrinsic fluorophore. For example, fluorescence protein, which is actually, at least the very first GFP was natural, so that means a protein that you find in nature. Then it was also modified. But here is used in this context is used as an extrinsic probe, and the nanoparticles, like for example the quantum dots. And in this case, you have to take care that the dimension of the probe it increases quite a lot in comparison to the organic dye. So yes, fluorescence protein, as I said, the first one that was discovered in the jellyfish was GFP. That he has a natural fluorophore due to the interaction of three amino acid inside, and is used actually as a marker, so that it's used as an extrinsic fluorophore. In which way? So then since is a protein, so when you express your protein of interest, you can modify the plasmid in the way that you can also have the gene coding for GFP. So in this case, you will get basically a fusion protein containing the protein of interest and the GFP, and your protein that maybe was not fluorescent, but then with the GFP close, it get also fluorescence, and you can visualize it. And this is an advantage because you can do this also, express this system in organism, in cell. And so that is a staining that is used very much in cell to monitor colocalization and so on, and also interaction. Okay, that is the basic method to use fluorescence protein. And since it's has quite some advantage and many applications, then after the discovery of GFP, there were a lot of studies to modify just with mutant to modify GFP. So there are several mutants of it in order to change the color of its fluorescence. And so here you have the example. So again, practically the same procedure that was done in organic probe to try to get all the color possible, and maybe increasing the stability, increasing the brightness, improving the quantum yield. So yeah, making, let's say, a fluorescence protein customized basically. Quantum dots are semiconductor nanocrystal, and the spectra depend on the bandgap size, so semiconductor, which depends on the size of the crystal. So the color is really dimension dependent of the quantum dot. And so you see the dimension, as I said, so they are quite large stuff. So the advantage of quantum dot are some spectral characteristic, like long lifetime. They have quite high brightness. They have narrow emission band and the broad excitation band. So it means that you can excite them with different wavelength, which can be an advantage and sometimes also not, because you maybe cannot be selective, and so it depends on what you want to do. And so that's it's known to know the characteristic of your probe, because then you choose the one that is appropriate to your system. That is clear. Okay, so, and about choosing, these are some criteria to choose the fluorophore that is adapted to your system. So what you have to go and have a look, you have to first of all see where we can excite and where it emit. So if it can correspond, can help you to investigate the system of interest or not. So then normally to have also a larger stock shift is an advantage to separate the excitation and the emission band. Molar extinction coefficient is an important characteristic. The fluorescence quantum yield, you want to use a probe that has a good signal in general. Lifetime also, if it's long enough to monitor your mechanism, in case you want to do some lifetime experiment. The brightness is actually a parameter that is often used to characterize probe, and it's combine the extinction coefficient and the quantum yield. So it's really a product of the extinction coefficient and the quantum yield, and that therefore is a value that has the unit of the extinction coefficient, since the quantum yield is without unit. And here, for example, some example of common chromophore, where all these characteristics are listed. And in particular, you can see that you have the brightness at the end, and this brightness it can be achieved, of course, with a large quantum yield. That's the case of fluorescein, which is one of the most fluorescent dye, almost one, you see. Or with a high extinction coefficient in case of cyanine dye. So that means that you can compensate if the quantum yield is not so good, but you can compensate with the extinction coefficient, because you say, okay, the quantum yield is not so good, but you easily excite, so it's more easy to excite it, and so then at the end you have anyway a good signal. Instead here you said, okay, it's less easier to excite, but the probability of excitation is lower, but as soon as he's excited, he has a large quantum yield, and so at the end the signal is also good. So so it's in this way that the story has to be seen. Okay, so fluorescent probe, for what we are using it, actually at the end, to get information, and these information are in our system of several kinds. So we want to localize a specific region, we want to observe how our system responds to physical chemical changing in the environment, pH, viscosity, polarity. You want to see interaction practically, respond to the presence or a change in concentration of a specific analyte, molecular mechanism in general, monitor reaction like enzyme activity, consider the fluorogenic probe I was talking about just before. And this we may want to do it in vitro, but also in a cell, and can be done also in vivo. And of course, there is a better probe for one or the other. So then it's one has to choose the appropriate one based on the parameter and that we just listed, and the characteristic, the chemical characteristic of the probe. Because that is actually the basic constituent of a fluorescence probe are three, actually two, and the third one is on depends on the molecule that you want to label with your fluorescence probe. So practically, a fluorescence probe is composed by a targeting unit and a sensing unit connected by a linker. The senses unit is your fluorophore, which is adapted to report on changes in the microenvironment. And that is the fluorophore. The targeted unit instead ensures the localization of the probe. And so it contains the reactive group that have to be adapted to the reactive group on the macromolecule to be labeled. So that is fundamental, because you want that your probe is where you want to put it. And so here is just an example for in case of proteins. So often are the reactive group are the amine or the thiol, and we will see, but that is schematically, that is the labeling, how labeling has to be done. And of course, important story is that the labeling principle should not alter your system. And in particular, since we are interested in biological activity, we should not alter the biological activity of the molecule. That's why several control has to be done when you change your system adding a probe, because a probe, yeah, is a good, it makes your system visible, but is something that was not there before, and you have to be sure that it doesn't change the system. Yeah. Okay, so then if you want to see, that are some the classification of fluorescence probes. So we can so here very schematically. So here in the middle, consider that is the events. So that says that you want to study. So that can be an analyte, that it can be a mechanism. So that is a more abstract, and you have your probe, you see, with the sensing unit and the targeting unit. So in this way, you can have three kinds of probe: the on-off. So that means the probe that was fluorescence before, when it's getting when it's observing the event, getting contacts with the event, it's is quenched. The on-off, which in general are the one preferred, and they are called also fluorogenic, because it's nicer to see a light, let's say, to switch on light on your system more than to decrease a signal that maybe it was already not so high before, but it depends. So both are used. And also you can the events may change the wavelength of the emission, so of your dye. So fluorogenic response is the one that is most, let's say, commonly used in the probe, and one mechanism that allowed this fluorescence enhancement is the photoinduced electron transfer. Just to give an example, so here, so chemist that want to design a probe, so actually has to know these things, because he has to design the chemical scaffold of the molecular scaffold of the molecule in order to favor such type of mechanism. And so here you have, for example, two examples. In this case is the electron, so photoinduced, so that is a consequence of the excitation, is transferred either from the recognition group to an excited acceptor, and or from an exciter donor to a recognition group, resulting fluorescence quenching. So then, as you see here, so then that is the case of the recognition group, that is the fluorophore, which is excited, and then the electron goes here. So then actually the donor cannot emit because it cannot lose his excitation energy because the ground state is already occupied by the electron. So that is how, for example, so now in this case, the photoinduced electron transfer is working. So you quench your molecule. And so the other way is in this way. So then is the excited donor to a recognition group. So then I'm just looking yeah, okay, so then the electron goes here in the lowest unoccupied molecular orbital of the recognition group, which cannot, and then since the ground state is occupied, it cannot go down anyway. So then again is a quenching. It doesn't emit because it goes here and it stays there. Well, this is the way in which, for example, this very known fluorescence probe is working. That is fluorophore, which is a calcium probe. So it's detecting the presence of calcium. And so you see, in this case, that is the recognition group, which is chemically done for coordinating a calcium ion when is there. So, and then you have a photoinduced electron transfer. Now, so in this case, from the recognition group to the excited donor. And so we are in the first case here. And so then the donor is non-fluorescence. But when the recognition group coordinate the calcium, the photoinduced electron due to the interaction with the calcium, so then the electron cannot be transferred anymore. And so this mechanism is blocked, and the fluorophore show its fluorescence. And so that's the way in which we detect, there is calcium there. Okay, then just the few minutes that are lacking, just to see also another other way in which we can get or change fluorescence in fluorescent probes, and how these mechanisms are practically telling us for what this probe is useful for. So, for example, here we have the isomerization of the cyanine dye can make this cis-trans isomerization. And the isomerization results in a high degree of non-radiative decay, which reduces the quantum yield of the dye. So that's so then somehow structural feature may affect the fluorescence of the dye. So just the proof that that is a analog, let's say very similar fluorophore, where the possibility of isomerization is blocked, and this is quite brighter than this molecule. Because here you basically lose energy, not only by fluorescence, but by the structural change in the protein. And here not. So that was an example how the isomerization may affect the fluorescence of the molecule. And now let's see how this can be translated in a fluorogenic response and for what is useful. So that is the case of molecular rotors that are di or this type. So where the fluorophore is actually the body pi, and so there is a rotation along this bond. So there are fluorophore that could form non-emissive twisted intramolecular charge transfer. So upon photon excitation. And so that means that you have more or less fluorescence according to the rotation. And in viscous environment, the rotation leading to the formation of this intramolecular charge transfer becomes hindered and the fluorescence intensity increases. So it's something like here that we have hindered the isomerization, and so then the fluorescence can increase. And so that's is. And so probe of this kind, the molecular rotor, are commonly used to study the viscosity of, for example, of biological membranes or local region in the cell, so the local viscosity, the micro viscosity in some system. And so that is what I wanted to say. So that's according to the process that can affect fluorescence, you know for what your probe is useful for. That's the message here. Okay, so I see that I reached the time. So I guess that you prefer like this, also because there is a session afterward that I stop here, and I just continue tomorrow, and we go on on fluorescence probes. Is fine?
Yes. Yeah, perfect. So any more questions from the students? Hello?
Yeah. I hear some interfering noise.
Simran?
Yes, sir.
Yes. So is there any question?
Okay. Thank you so much, ma'am, for an informative talk. I wish you all had an insightful lecture.
Okay. So yeah, I hear actually it's interfering. I don't know.